Hello everyone, and welcome to the section 2 of MSAT Achieve Biology series. This is section 2.2, which is explain transcription, post-transcriptional modification and translation, and relate these processes to gene expression in prokaryotes and eukaryotes. Okay. Under this, we first need to know what do we know about the central dogma of biology. The central dogma of biology is very simple. The information flows in the direction from DNA to RNA to protein. Okay. For example, if I have a purple flower, this purple flower is due to a protein that makes this purple pigment. So how do we get this protein? This protein would be produced from the DNA, which will be present in the flower itself. Okay. So the DNA has some information which helps to make protein. So please listen here very carefully. How can I put this the whole thing here? Just imagine you have to make a cake. So if protein is a cake that you want to make, and you have a recipe which is written in a language which is unfamiliar to you. For example, the recipe is written in, say, Japanese. I don't know Japanese, okay, but I want to make cake. So what will I do? So this recipe is in the form of Japanese. So what will I do? First, I will translate the recipe into something which I can understand. So I will translate the language of DNA, which is ATGC, into the language of RNA, which is AUGC. As you know, T is present only in DNA, and U is present only in RNA, right? Thymine in DNA, uracil in RNA. Okay, so now when I have this language encoded into a language which is familiar to me, I can start making my cake. Okay, so this is what the whole process is. So making mRNA from DNA is called transcription, and making proteins from mRNA is called translation. Okay, now before we start, we have to know the three different types of RNA which we will be talking about in this unit. First is the mRNA. It does nothing but just translates or encodes, or rather, what would I say? It actually interprets information which is there in the DNA. The information in the DNA, it translates it, or it changes it into a language of RNA, which is AUGC. Okay. Then you have something called rRNA. This rRNA is basically, R means ribosomal, so it helps you to make a ribosome, which of course you know is the machinery for protein synthesis. It makes proteins. So if you have no ribosomes, then you cannot make proteins. And then you have tRNA. This tRNA is called transfer RNA. The whole purpose of it is to carry amino acid to the ribosomes, which we will see in translation, these two parts. But this one, mRNA, we will see in transcription. Okay, so here, how does the transcription thing happens? Please remember, DNA is a double-stranded molecule. One strand runs from 5' to 3', and the other strand runs from 3' to 5', since they're antiparallel to one another. Now, mRNA or RNA that you're making is single-stranded. So you cannot make use of both the strands of DNA. You can only use one strand of DNA. So the strand of DNA which you use for making mRNA is from 3' to 5'. We never use the 5' to 3' direction DNA for making your mRNA. We always use from the DNA which is running from 3' to 5'. This is also called your template DNA, or antisense DNA, or non-coding DNA. There are multiple names for that. Okay, and the mRNA which you make after transcription will be in a direction which will be opposite to your template DNA. That means if you have 3' here, your mRNA will have 5' here. If you have 5' in the DNA, your mRNA will have 3' here. And rest, just write the complementary sequences. T pairs up with A pairs up with U, C pairs up with G. Why are we writing U here and T here? Because this is a DNA and this is an RNA. Okay, wherever you have A, you will write U here. Okay, because in RNA, please be very careful while you are encoding the answers. Okay, again, So DNA is a double-stranded molecule. We do not use both the strands of DNA. We just use the strand of DNA which runs from 3' to 5' direction, and then make an RNA or your mRNA, which is your mid compound here, which will help, which is a product of transcription rather. And the product that you get here after transcription is not your final product. We will see that it needs some processing, which we will study in mRNA processing a little later. Okay, so here the 3' to 5' again is your template DNA, and your RNA, the mRNA, would run from 5' to 3'. The same information, I'm repeating it multiple times because this information is important. The next thing you have to know is the location of these processes in a eukaryotic and a prokaryotic cell. In eukaryotic cell, we have a nucleus. In prokaryotic cell or a bacterial cell, we don't have a nucleus. So in bacterial cell, where we don't have a nucleus, both your transcription and translation takes place in the cytosol or the cytoplasm. Okay, but in eukaryotic cell, we do have a nucleus. So your transcription and the processing of your mRNA will take place inside the nucleus. And as I told you, after transcription, the mRNA that you made is the primary transcript. It's not the final one. You have to process it further to make your final mRNA. And once this processing is done, after that, it will come out of the nucleus. So your transcription and processing is done inside the nucleus. And then this mRNA will come out in the cytoplasm or the cytosol. And then it will go and work with the ribosomes to make your proteins. And that process we know is called translation. So what is this RNA processing that we are talking about? Please remember, it happens only in eukaryotic cells. That means plants and animals. It will never happen in bacterial cells. Okay, what happens here? In mRNA processing, we are actually doing 3 things to the mRNA. We add a 5' cap, we add a 3' poly A tail, and we do some splicing or cutting of the mRNA. So what do we do exactly? So this 5' cap, why 5'? Because I am putting a cap at the 5' region of the mRNA, and this cap is actually made up of a modified guanine nucleotide. And then at the 3' end, I have multiple A's, which is your adenine. So this is called a poly. Poly means multiple A at the 3' end. And both, what is the purpose of putting a cap and a tail? So they are there actually for protecting your mRNA. Why do I need to protect my mRNA? Because it has to travel all the way from the nucleus to the cytoplasm, and along this journey it might encounter a number of enzymes that might harm it in the way. So I need my mRNA protected because I need to make proteins. So this cap and the tail protects your mRNA and prevents it from being broken down by enzymes. Okay, and the journey that it is, and it also helps in other processes, makes the transcript more stable, but you're not supposed to learn those things in details. After putting a cap and a tail, the next step is cutting or splicing your mRNA. Why I'm cutting that? Because the mRNA that we made has some good sequences or protein coding sequences called exons and some useless sequences called introns. I don't need introns in my system. Why? Because introns, they don't code for genes or they don't code for proteins. So if they don't make proteins, why do I need it? My whole aim is to make proteins in translation. So what will I do? I will cut the introns or the useless sequences and then simply join the exons together. Okay, so now this is your final product after splicing, which is known as mature mRNA. This was all about transcription and your mRNA processing. Now the next step is translation. Before we go into the next step, you should know what are the characters you will come across in translation. First one is ribosomes, because they are the cell organelles that makes proteins for you. Ribosomes are made up of 2 subunits, the larger subunit and the smaller subunit. The larger subunits has 3 sites: E site, P site and A site. And we will see the purpose of it in a little later when I'll be explaining translation to you. Well, the A site is basically for, we will see the binding of the tRNA molecules where it's bringing the different amino acids. The P site is where the peptide bonds between the amino acids will be formed. E site will be where the tRNA will leave after it has delivered the amino acid. We'll see all that in a while. The next thing is the transfer RNA or the tRNA. The transfer RNA looks something like this. It has a 3' end and a 5' end too. But this 3' end, it carries an amino acid. Okay. And this amino acid is complementary to the sequence on the mRNA. For example, you have GAG. So this tRNA will carry the amino acids which has this sequence GAG here. Now, at the other end, the tRNA has something called anticodon. Anticodon is complementary to your mRNA sequence here. So if you have G, I have a C. If I have A, a U, G, and D. But the amino acid it will carry, it's not CUC. The amino acid it will carry is GAG here. Please remember that. It will never carry the amino acid for these letters. It will carry the amino acid for the letters on the mRNA. Okay. Now, so what happens? The first step which happens in translation is the first amino acid. The first amino acid is always methionine. Okay, methionine, which is AUG. So the first thing that will go is the mRNA, and your tRNA will start scanning your mRNA for a sequence which has AUG in it. The moment it finds AUG, what happens? The tRNA will come and bind to the AUG sequence. So it's coming and binding to your P site. This is your P site. The first amino acid will always bind to the P site, and the other amino acids will join from the A site, never from the P site. Okay. So once the methionine joins here, the tRNA joins here, taking this is the methionine that it's carrying, then the next tRNA will come and occupy the A site. Okay, it has a different amino acids. Now this tRNA will deliver the methionine and its job is over. It will go to the E side and it will be removed from the system. And then the tRNA which was in the A side will move to the B side. See, now this pink one from A side, it's in the B side. Now the next tRNA with the new amino acid will come and occupy the A side. And this process will keep on continuing till they encounter something called a stop codon. The moment they have a stop codon, your polypeptide or the protein making will be complete and it will be released. Okay. Now we will quickly do some exam questions. The first one that we have here is, these questions are from your MSAT exam. So these questions under this is, which process results in the synthesis of mRNA transcript? So synthesis of mRNA, which process do you make mRNA? You make mRNA in the process of transcription. Okay, so the answer is transcription. The next thing is they are giving you this diagram. See, this is the DNA, you're making mRNA. So what is this process? Number 1, transcription. What is number 2? See, what is now happening in number 2? From 420 nucleotides, you come to 120. Something is missing. You're cutting something. What is happening in 2? You're actually removing something, right? So they're asking what is happening in step 2. So what is happening? Introns are being removed, right? So we don't need the introns. Introns are useless sequences. We just keep the exons. This is what is happening. Okay. And the other, as you will see, the other options here doesn't make any sense. So we are not even reading them. Now the next one here is, they have given you a sequence for DNA. Now they're asking you what will be the mRNA sequence. As I said, if you have a 3', simply choose a 5' for your mRNA, 5 to 3. So your mRNA is 5 to 3. And since it's an mRNA, never you will have T, you will have U. So you will add your, so it will be 5 to 3. And every complementary sequence, that's C with C, G, G, and then instead of with every A, you put a U. And your answer would be answer number D. Coming to this one, what is the process of protein synthesis? We know from DNA to RNA to protein. The DNA to RNA is called transcription and the RNA to protein is called translation. So the option C is the correct answer here. And okay, and coming to this question, this diagram shows part of central dogma of life, which we know. What is the central dogma of life? DNA to RNA to protein. DNA to RNA is transcription, and RNA to protein is your translation. Okay, so what does they say? What is the process shown here? Of course, there's a ribosome, there's an mRNA, there's protein being made. What do you think it is? Is used to produce another copy of DNA? No, that's DNA replication, right? Copies the genetic information from DNA and carry it to the cytoplasm. This is mRNA transcription, right? Uses mRNA sequence to produce proteins. Yes, this is what we do here. We use the mRNA sequences to make proteins. So this is actually translation, which is happening in the picture. And then finally, uses DNA and mRNA to produce ribosomes. We never use DNA and mRNA to produce ribosomes. We use rRNA, ribosomal RNA, and proteins to produce your ribosomes. Okay, so the correct answer here would be option number C. Now coming to the final question for this, they've again given you a diagram and something is happening, and we can see what is happening. You had exons, which are these deep color sequences, and introns, the lighter ones, and now something happened. After that, we just have the deep color sequences. So we know what this is. We know this is splicing, right? So we are splicing off the introns and we're just keeping the exons with us. Now, let's look at the options that you have. The first option, it takes place in the nucleus. Yes, it does. mRNA processing takes place in the nucleus, and I have shown you the picture for that earlier. Also, let me show you again. Transcription and processing takes place in the nucleus. Okay, so for option number 1 is correct. Now coming back here, exons are removed by the end of the processes. Do we remove exons? Why should we remove the protein coding sequences or the good sequences? We don't remove them, we remove introns. Okay, so option number 2 is incorrect. Now option number 3, it produces an mRNA that is ready for translation. Yes, that's the whole purpose of splicing or mRNA processing. We are processing the mRNA to make a final mRNA which is ready for producing proteins. So this option 3 is also correct. Okay, so the option number 1 and option number 3 are both correct answers. So here I will write what will be the answer to this. Will be option number D. With this, we finish this video. In the next video, we'll be discussing some other topics. Thank you.